Hello, my name is Dr. Roderick L. Roll, and today we will be talking about sexual reproduction on the cellular level. Sexual reproduction requires fertilization between gametes. These gametes are called either an egg or a sperm. In this chapter, we're going to talk about how to produce an egg in a female and how to produce sperm in a male. The production of these gametes are called meiosis. The human somatic cell, also called the body cell, has 23 pairs of chromosomes. So a body cell is something like a bone cell. A body cell can be a liver cell. It can be a nerve cell. So all of these cells are referred to as somatic cells. A germ cell is a cell that will exist in the ovary of a female and the testicles of a male. So the majority of the cells in your body will have 23 pairs of chromosomes. These 23 pairs of chromosomes can be visualized through a karyotype. A karyotype is an ordered display of those 23 pairs. The organization of the pairs based on length, based on the centromere position, and based on the staining pattern will be achieved during this karyotype formation. In humans, we have 23 pairs of chromosomes. These pairs are called homologs or homologous chromosomes. It has the same length, it has the same shape, and it carries genes that control the same inherited characters. A inherited character can be something like hair texture or eye color. So like I discussed, the humans have 23 homologs, or they have 23 homologous chromosomes. In this image, I'm showing the pair of chromosomes, one red and one blue. In the picture below that, the chromosomes have replicated, so it's still referred to as a homolog. Here's another way to look at it. In the first picture, we have one chromosome. This one could have been received from your dad, or it could have been received from mom. This chromosome is going to go through a replication process during interphase. Remember, interphase has G1, S, and G2. During S phase, that chromosome will be replicated. It will still be linked together by a centromere. So in the picture in the middle, we have a duplicated chromosome. And then further on in the slide at the bottom, you'll have those two chromatids separate. When they separate, we will then call it two individual chromosomes. But in the beginning, we have one chromosome that is replicated, and it is joined by the centromere. Then that chromosome will split during anaphase to form two chromosomes. Human somatic cells, or body cells, will have 23 chromosome pairs. One of those pairs is going to be referred to as the sex chromosomes. If you are a female, you will have an X and an X. If you are a male, you will have at least one Y. The other 22 pairs are called autosomes. Most human cells are diploid. Remember, the body cells are also called somatic cells. These somatic cells are going to have a father's copy of each chromosome and a mother's copy of each chromosome. So we're going to call that paternal copy for the dad and maternal copy for the mom. Each gamete that you produce is going to pertain either a father's copy or a mother's copy. The gamete is either a sperm or an egg. The fusion of these haploid gametes is referred to as fertilization. When the sperm unites with an egg, you will create a zygote. A zygote is the reuniting of these haploid cells to create a diploid cell. In this image, we see the reunion of the haploid gametes to create a diploid zygote. So in the haploid state for a human, the sperm only has 23 chromosomes and the egg only has 23 chromosomes. When you reunite those two gametes, you will then have a cell with 46 chromosomes. Let's run over meiosis really quick. You will start off with interphase, where you will have chromosomes that will go through a replication phase. Remember, interphase is separated into G1, S, and G2. So the homologous pair that we see in this image is going to double itself. So we have one chromosome pair, or we have two chromosomes in this first image. In the bottom image, we still have one homologous pair. Now, the homologous chromosome separates during meiosis 1. More importantly, it is anaphase 1 of meiosis 1. These haploid cells are going to then go through meiosis 2. Why is those cells haploid? They're haploid because the blue X versus the red X, they're only paternal copy or a maternal copy. For it to be diploid, the X would have to have one red line and one blue line. So therefore it's haploid. So now these haploid cells are going to go through meiosis 2 and separate that homologous chromosome into chromosomes. So during meiosis, haploid gametes are produced. They're going to be produced in the gonads, either the ovary for female or the testicles for a male. Interphase is then going to be followed by two consecutive meiosis divisions. We're going to call that meiosis 1 and meiosis 2. In between meiosis 1 and 2, you will not see another interphase. If so, you would double the chromosome number again. So no, there's only one interphase, and it is before meiosis 1. In the next few slides, we're going to go through meiosis. So the first thing that has to happen is we're going to have to increase the cell size. We're going to have to double all of the organelles. We're going to have to increase the number of mitochondria. We're going to have to increase the length of the endoplasmic reticulum. We also are going to replicate all of the genetic material. That occurs during interphase. Then we're going to go through meiosis 1. So we go through prophase 1. Then we're going to have metaphase 1. And then we're going to have anaphase 1. We're going to go through what happens in each of these stages. So first, in prophase 1, the homologous chromosomes pair up. The homologous chromosomes can also be called a tetrad because it will have 4 little chromatids that will be called a tetrad. Notice that some genetic information is going to be exchanged during prophase 1. We're going to create what is called a chiasmata in the tetrad. We're going to explain this in much more detail during lab. After prophase 1, then we go to metaphase 1. The tetrads are going to move around and line up on an imaginary plane called the metaphase plate. How it moves is through the microtubules. The microtubules are produced from this centrosome, and it will attach itself to the kinetochore of the tetrad, and it will be moved. From metaphase 1, we will now go to anaphase 1. This is when the tetrad separates. So it's 4 chromatids joined together. They're going to split apart, and now we're going to have dyads, or a duplex that is going to be formed. So you're going to pull apart the 4 chromatids, and now you're only going to have 2 chromatids that are still going to be called the chromosome. So homologous chromosomes separate during anaphase 1. Now we're going to go to telophase 1, where the nuclear membrane begins to reform. Then we're going to have cytokinesis, where we're going to pinch off the cytoplasm. Now we have 2 individual cells that are both haploid. The next stage is not interphase. We're going to go straight to prophase 2. Prophase 2, the nuclear membrane degenerates, the centrosomes begin to move to their respective poles. The next stage is metaphase 2. During metaphase 2, the spindle fibers or microtubules are going to attach to the chromosome. It's not a tetrad now, it's a duplex or a dyad. So now the spindle fiber is going to attach to the chromosome, and then in the next phase the chromosome is going to separate. So during anaphase 2, the chromosome that was composed of 2 chromatids will now separate. Once the chromosome separates, each chromatid is no longer called a chromatid, it is called a chromosome. So those chromosomes move to their respective poles. Now we're going to move to telophase 2. Once those chromosomes are to their respective poles, the nuclear membrane will reform. Then you will have cytokinesis again. So this is a summary of meiosis 1 and meiosis 2. During lab, you're going to take beads and demonstrate this process. Now we're going to compare and contrast mitosis to meiosis. The original cell has 6 chromosomes, 3 blue and 3 red. Let's look to the left. To the left, we're going to go through mitosis. Those 6 chromosomes are going to replicate. The next thing that occurs is those 6 replicated chromosomes are going to line up on an imaginary plane. That is called metaphase. The next thing that will happen is you're going to split the cell in half. you're going to pull those 6 replicated chromosomes, you're going to split them, and now in each subsequent cell, you're going to have 6 chromosomes again. This is going to duplicate the original image. So now let's go back to our original image, and now we're going to go to the right. So meiosis, we're going to also replicate all of the chromosomes. But instead of the chromosomes being randomly distributed within the cell, they're going to now be attracted to their homologous pair. So now you won't see a duplex, you're going to see tetrads in meiosis. Now instead of random chromosomes lining up on an imaginary plane, you're going to have tetrads lining up on an imaginary plane. Then you're going to go to anaphase, where we're going to split this tetrad. You're going to split this tetrad in anaphase 1, and then you're going to take it through meiosis 2, and in the end you'll see that you have half the number of what you started with. The behavior of chromosomes during meiosis and fertilization is responsible for most of the genetic variation that arises in each generation. 3 mechanisms contribute to this genetic variation. The first one we're going to talk about is independent assortment of chromosomes. During this meiosis process, these chromosomes are going to independently arrange however they want. So we're going to discuss that. Then we're going to talk about crossing over. Then we're going to conclude with random fertilization. But all 3 of these are going to create genetic variation in the population. This is why all of us look very different. There are no individuals that are genetically the same, except maybe twins, but we'll discuss that later. So the first one we'll talk about is independent assortment. So during this process, chromosomes move independently of one another. In meiosis 1, there's a 50% chance of a father chromosome being in the first daughter cell. There's also a 50% chance that the mother chromosome will be in that first daughter cell. So this is random. So let's look at the picture I tried to draw to illustrate this phenomena. In that daughter cell A, there's a 50% chance to get those 2 blue chromosomes or to get the red chromosomes. This is random. If you happen to be created from the daughter cell A, then you would have chromosomes that came from your father. So this is showing probability. One probability, you get all of the blue chromosomes on one side of the cell. Probability 2, you might have 1 blue on the left and 1 red on the left. The next step in this process is to separate the homologous chromosome. In this process, you see that in probability 1, the cell on the far left will be all dad. Probability 2, the cell on the left will be 1 chromosome, 1 replicated chromosome from dad and 1 replicated chromosome from mom. Continue this process down, and you'll see the different variations in the daughter cells. If one of those daughter cells is the sperm, in probability 1, the far left, combination 1, you're going to have chromosomes that came from your dad. So all of those genes from dad, you're going to express. If it was combination 2, and that was the sperm that produced you guys, then that will be all mom's chromosomes. Versus probability 2, you would have a variation. You would have one mom chromosome and one dad chromosome for each of the daughter cells. So the number of chromosome combinations possible when they assort independently into gametes are 2 to the n power, where n is the haploid number, or n is the number of chromosome pairs in question. So let's do a couple examples so I can make sure you clearly understand this process. So how many combinations are possible for 2 chromosome pairs? This is how you would perform this question. How many combinations are possible for 2 chromosome pairs? So the total number of chromosomes is 4. So the haploid number is 2. So 2 to the n power, or 2 to the second power, or 2 times 2 equals 4. So the answer is 4 total possibilities. To illustrate this, or to try to show this, I drew it. So you take the tall red and tall black, and the short red and short black, and that is the chromosome that is in the cell. So we're asking, well, how many variations do we have in reference to the gamete? Someone said this reminds them of the FOIL method. So you take a line and you draw from the tall black or big black to little red. That's one gamete. Then you go from big black to big red. That's another variation. Then you go from little black to little red. That's a variation. And then little black to big red. So each one of those are probabilities or possibilities that will be achieved when you produce a gamete. So either one of these gametes can create the next offspring. So let's try another one. How many combinations are possible for 3 chromosome pairs? So same concept. So now, instead of doing 2 times 2, now you just do 2 times 2 times 2. So here's the answer. How many combinations are possible for 3 chromosome pairs? The haploid number is 3, so then therefore you replace n with 3. Therefore that will be 2 times 2 times 2, which is 8. 8 total probabilities. Either one of these probabilities could create a new offspring. This is why offspring will not be genetically the same. Here are the answers to the rest of them. Now that we know how to figure out the probabilities, I would like you to answer this question. How many possible chromosome combinations are there for the corn gamete versus the fruit fly gamete? I'm about to show you the answers. Push pause if you're not ready. The answer for the corn gamete is 1024. The answer for the fruit fly gamete is 16. The most common mistake is when people look at the chart and see the diploid number, They forget that they have to use the haploid number. So fruit fly you use 4, and for corn you use 10 to replace the N. How about for humans? Humans have 46 total chromosomes. Again, that's the diploid number. You have to take the half. Half would be 23. So for humans, N will be 23. So the possible combinations, when you put 2 to the N power, and N is 23, will be 8.4 million per gamete. So these are different variations or different possibilities of gametes. One of those gametes created you, or one of those gametes came from your dad, and one of those gametes came from your mom. So therefore, there was 8.4 million different types of sperm that your dad produced, and 8.4 different types of eggs that your mom produced. So the number of diploid chromosome combinations in a human zygote is going to be 70 trillion. If you take into consideration your dad probabilities versus your mom probabilities. So again, this is why you are so unique. This is why you are so special. There's no other that resemble you. The second thing that created you, that made you so special, is crossing over. So crossing over produces recombinant chromosomes. So now the chromosome can change. So this means you're going to combine DNA inherited from each parent. So if we look at our image, we have a blue chromosome and a red chromosome. This crossing over event will only occur between homologous chromosomes. So let's look at the picture, and you see we have an orange and a yellow chromosome that is replicated. Since they are homologs, they are side by side. They're so close that the two closest chromatids can touch. And when they touch, they can cross over and wrap around each other. And then when they break apart, you're going to have the genetic exchange of information. So look at the image to the far right and the tetrad. Some of the yellow chromatid has been exchanged to the red side. This is going to create variation. If your chromosome is the one that's on the far outskirts of the tetrad, then you're going to be all mom or all dad in reference to that chromosome. If you are the chromosome that's in the middle, you're going to be a variant. You're going to be different. Your chromosome is going to have most of mom with some of dad, or most of dad with some of mom. When does this occur? This occurs during prophase 1. So during prophase 1, we actually have 5 stages. We have leptotene, zygotene, pacotene, diplotene, and dikinesis. So during prophase 1, we actually have 5 different stages. During those 5 stages, you're going to have the genetic transfer of information between sister chromatids. Let's look at it in much more detail. So number 1, non-sister chromatids of a homolog will break. Look at the image and you'll see the red line has broken for one chromatid, and the blue line has broken for one chromatid. Number 2, the synaptonemal complex joins the homologue. That green circles are like a molecular glue that holds together the tetrad during prophase 1, during those 5 stages. So the tetrad is being held together. We've already broken the 2 closest chromatids. Now the glue is holding it together. The next step is number 3. The exchange of genetic information occurs through a crossover event. So now the broken chromatids are going to be soldered back together, but red didn't get joined with the other red. It got joined with the blue. So that is the crossing over event. That is the exchange of genetic information. Number 4, now the chiasmata is going to hold the homologous chromosome together through the rest of prophase 1, through the rest of metaphase 1, until they separate. Once they separate, now if we look at this bottom image, the top blue is all dad. The bottom red is all mom. The 2 closest chromatids that are in the middle, they're going to have some dad mixed with some mom and some mom mixed with some dad. So in humans, an average of 1 to 3 crossover event will occur per chromosome. The third and final thing we'll discuss that created such a wonderful human being is random fertilization. In our country, any sperm can fertilize any unfertilized egg. In other countries, it's not so cut and dry. You have arranged marriages. Back in the day, you had slave masters that wanted to have a genetically superior individual, so they would take people that had certain traits and cross them with other individuals with certain traits. So that doesn't happen as frequently now. Now anybody can fertilize anybody. So that is also going to create genetic variation. So what makes you so unique is a combination of all 3 of these. You have random fertilization, you have crossing over, and you have independent assortment of the chromosomes. All 3 collectively makes you a very unique individual.